All right, in this video, video 9 of chapter 4, we're going to look at translation, how we're taking this messenger RNA copy of the gene that we have and translating it into a useful set of information that we call a polypeptide, which will turn into a protein. Again, chapter 4, this is toward the end, we're looking at translation. So when we're looking at DNA, each group of 3 bases, right? So remember, DNA is the 2-stranded helix, and there's bases that connect each other through the hydrogen bonds. Each group of 3 is referred to, on this it says a codon. Now I've heard it called triplet before, but again, in this book it calls these 3 a codon. Now each group of 3 represents an amino acid. All right, so as we're going through, we'll kind of talk about that. Now, when we are talking about transcription in the previous video, we said that it happens in the nucleus. It's where the DNA opens up, messenger RNA makes a copy, and remember, it copies all these bases down, and as it makes a copy of those bases, the only thing that's different between the A and T and the C and G of the DNA codes, that there's no T in RNA, no thymine. It is uracil, or a U. And so once we can make this copy, the messenger RNA is going to go out and find a ribosome, because the ribosome is the place where we synthesize the protein. And at that point, we go through what is called translation. It is in the process of converting the genetic code carried by RNA into a sequence of amino acids that can become a protein. So I kind of say that the translation is making it useful. If I gave you, we've got a test, let's say we have a test next week, and I gave you before the test, a week before the test, the test written in some foreign language. I don't know, a Mandarin Chinese or Danish or whatever. If I'm giving you something and it's in a different language, it's not useful. You will have to go through the process of translating it in order for it to be useful. And that's kind of what happens at the ribosome. So we looked at this picture in the previous video, and I said that this over here was called transcription, writing it down, and this shows the messenger RNA leaving through that nuclear pore, finding one of these guys, which we call the ribosome, which is basically the kitchen for making proteins. And again, as it goes through, we're going to see how this moves through this ribosome, 3 sections at a time, which we said H3 represents an amino acid. And on the messenger RNA, those 3s are called codons for sure. Now, what happens here is as this messenger RNA is being pulled into the ribosome, all right, there's going to be groups of 3 amino acids that will sit into a little spot. So if this is the ribosome, imagine that it has this little skylight, all right? And so as this messenger RNA is being pulled through, it's going to stop just long enough underneath this skylight that there's going to be another type of RNA called transfer RNA, which is going to look into that skylight and see those 3 bases that we call that represent an amino acid, but those 3 we call a codon. And the transfer RNA will make a copy of it, and we call that copy the anticodon. Now the transfer RNA will then leave and get the amino acid that is corresponding to that anticodon. So imagine that I've got this box of Legos over here, and inside this box I've got 20 different types, 20 different colors of Legos. And so we're going to see in just a minute, there's 20 different amino acids that build up proteins. So what is really happening is the messenger RNA carries this alphabet of 3 in these bases. As it goes through the ribosome, each individual 3, and we call them codons, will stop underneath this skylight, and transfer RNA will make a copy of it called an anticodon, go to the box of Legos and pick out the Lego that corresponds to that the color of Lego that corresponds to that anticodon, bring it back and start placing it in position. And then the messenger RNA is going to be pulled up to the next three, and then the next three are exposed under this skylight. The transfer RNA goes and gets the next, makes an anticodon, goes to the box, gets the next color, and it continues to do that until it grows. Now, think about this. Most amino acids are hundreds, or most of proteins are hundreds of amino acids long. So if I'm talking about something like hemoglobin, which is a couple hundred amino acids long, you're talking about times 3 the number of bases that we're talking about. So each amino acid, if it's 300 amino acids long, that means that there are 900 bases, and they're read in the little groups of 3. And so it's going through making these. Now, again, like I said, there are 20 types of amino acids. So imagine 20 different colors of Legos in our little box. There ends up being 64 possible codons on RMA, which just means there's 64 different orientations of that. And so many of the amino acids will have several different types of codons that will represent it. Now, when we're looking at this, there is an initiation codon, which means that's where we start, and a stop codon, which is where we finish. And it's all, you don't have to know them, but I would hope you know an initiation codon is the start and the stop codon is the finish. So as it's going through, there's going to be this process of reading it and taking it from the codons, the groups of 3s on messenger RNA, and through the process of transfer RNA, making the anticodon and bringing the amino acid into place, now I'm translating it into a useful image, because proteins are links of amino acids. This is the 20. If you ever have kind of like problem sleeping, you might want to go through these, but I'm not testing you on these, but if you ever get into more advanced biochem, you'll have to know them. Now, ribosomes, these very special organelles, are actually composed of ribosomal RNA, or another way of saying rRNA. Now, there are 2 unequal subunits, but they are both rRNA. And so binding tRNA, transfer RNA, and mRNA, messenger RNA, occurs in association with the ribosome. So it's these 3 kind of working together to make this happen. So as the amino acids are brought into place, again, they're joined by what we call a peptide bond. And since we're adding them to it, we are doing this by the process of dehydration synthesis, right? Synthesis type reaction, building, and we're doing it by removing water. So every time it brings the transfer RNA brings that amino acid in, I'm going to remove water, and I'm going to cause those amino acids to form a peptide bond. This is kind of showing the process as it's pulling it through. Again, there would be, this little skylight right here. And so as it's moving through, it's going to place the next amino acid in the chain, and the chain will just keep growing until we have a polypeptide, and then it will be packaged as a protein. So these are the steps. Transcription, which occurs in the nucleus, has messenger RNA that's going to get in the middle of the 2 strands of DNA and make a copy of one of the strands. Big difference is when it's making a copy, it does not use thymine. It will use uracil in its place. The messenger RNA will pass through a nuclear pore and get into the cytoplasm. At that point, we'll look for a ribosome so that we can go through translation. So translation, again, happens in the cytoplasm. When the messenger RNA binds to the ribosome, it'll start pulling that messenger RNA through 3 bases at a time. That's a group we call a codon. And at that point in time, transfer RNA is going to make an anticodon, go to attach to the ribosome that is associated with that anticodon, and bring it back into place until a complete protein is created and released. And then messenger RNA, the ribosome, and the transfer RNA are all recycled to do this over again. Again, hope that makes sense. Don't over, I hope you know a lot of the basics, but don't, or a lot of the fundamental, more complicated parts, but I want you to make sure you know the fundamentals. Transcription happens in the nucleus. Messenger RNA makes a copy. Translation happens in the cytoplasm, particularly on a RNA, on a ribosome, and transfer RNA is what's going to take the codon from the messenger RNA, make an anticodon, and then use that anticodon to bring in the amino acid and make this a usable molecule that we call protein. I hope that helps, and have a wonderful day.